material. Keep in mind, I already wrote the exam, so I know it's going to be on, so maybe I would
Pay attention and shut.
pay attention. So, digestive system. Again, to understand it, it is best to kind of follow, right? The ingested food, what's happening over here, what's happening down here, what's happening over here, what's happening over here. Just follow the food down as it goes down. Name the structures that are involved. And it's usually like a 1 or 2 word thing, right? Digest proteins, digest carbohydrates, absorbs food, something like that, right? So we'll walk through these, right? But each region, right? The oral cavity, like the esophagus, basically, stomach, small intestine, large intestine. What's going on there? What are the salivary glands doing? What is the liver doing? What's the gallbladder doing? What's the pancreas doing? Right? As far as processing this food, that slide should be on your slide somewhere. But it, gives a very brief rundown, right? You should know the functions of all the major stuff for sure, right? As well as the name, like what's movement, forward movement through the tract called? A bolus?
Yeah.
What?
Aries.
Peristalsis, right? Whereas this moving kind of back and forth, breaking it down mechanically, like you see in your stomach, for instance, is segmentation. Right? Both of them have to do with the contraction of your inner and outer layer of your muscle layer of your digestive tube. Remember all those? Right? But also in general, just as far as the organization of your abdominal cavity, you got those serosa making up the visceral and parietal semosa form the peritoneal cavity. And then you got those extra structures, the mesenteries, right? That were kind of holding the intestines, the back wall to each other. Other functions included blood vessels, nerves, and lymphatic vessels going through those. They also stored visceral fat, right? That's important. Subcutaneous fat, remember, is the stuff under your skin. Visceral fat is what you're getting from eating all these brownies right now. No. Visceral fat is that fat that's stored like it would be inside the mesenteries. That's like it's a big metabolic red flag when you got a lot of visceral fat, right? So that's where it's stored in the mesenteries. Not subcutaneous fat. All right? Subcutaneous is under the skin. All right, so for the stomach, right, esophagus, in the mouth, you have started to digest the carbohydrates with salivary amylase, right? And also lipids, but we'll just stick with the carbohydrates. In the stomach, it's going to be mechanical and chemical digestion, swishing back and forth. What sound does it make?
Ariel.
Thank you. Like that. It's swishing back and forth, it's squishing back and forth. It's mixing with the acidic juices, gastric juices, right? Hydrochloric acid and pepsin or pepsinogen is secreted by those 2 types of cells. Parietal cells are secreting, there's parietal and chief cells.
HCl.
Parietal is the HCl, hydrochloric acid. The chief cells are the ones that are secreting the enzyme pepsin or pepsinogen to digest proteins. Remember, pepsin is not produced by parietal. They're the chief cells. Right? In any case, those are the rugae that allows for expansion of the stomach. The cardiac sphincter prevents that acidic chyme from going into the esophagus. The pyloric sphincter allows the acidic chyme to be kind of let out a little bit at a time into the duodenum right here, right? So it's very acidic, and it's going to be dumped in a little bit at a time. That acidic chyme could be neutralized by sodium bicarb-like secretions from the pancreas. Okay? So there's your duodenum. The last stage of digestion is happening in the duodenum with the help of the gallbladder, which is like secondary, like it gets bile from the liver, and then it secretes the bile into there. That breaks down fat. That's emulsification. Right, of the fat, and then the pancreatic ducts secrete all those. That's going to break down proteins, carbohydrates, and lipids, like everything else. Right? So, mouth, carbohydrates, proteins, stomach, and then everything else is the finishing, the last of the digestion with the help of the liver, the gallbladder, and the pancreas. Okay? In the duodenum. Right? So that's the last stage of digestion. Right? And then once you move into the small intestines, the rest of the small intestines is your jejunum. The main absorptive area is the jejunum, you have those folds that are related to that, right? You got the circular folds, villi, you got microvilli, right? These layers of folds that massively expand what? Surface area, surface area, surface area, surface area, surface area. Right? Those are all surface area expansions. Right? Because we want to absorb every little last bit of that brownie that you ate. It wouldn't be better if you just pooped it out, right? And then you wouldn't have all that high blood glucose. But we're not designed for that. All right? So all throughout, whether you're talking about your stomach, your small or large intestine, you have those layers, right, that make up a tube, and typically, right, your epithelial layer, stratified squamous up here until you get to the stomach. Then it's going to transition to a simple columnar, right? Because when you look at this, why? So here, it's going to be stratified squamous because it's going through like a tight tube and you need protection. Protective stratified squamous. Once you get into here, it opens into a wide open space right here. And it's going to be simple columnar all the way through until the very end. All right?
Was there enough to be boiled?
No, it's just those two, stratified squamous or simple columnar. Right? And then that would be the simple columnar. And you'd have a lamina propria for the models. Those models also for the practical. There's that digestive model, everybody knows what I'm talking about. It's got these layers on it. The mucosal epithelial, the lamina propria, and the muscularis mucosa belong to the mucosa layer. Submucosa doesn't have any sublayers. And then the external muscular layer with the inner and outer layer, or circular and longitudinal. And then the serosa on the outside. Right? And this is going to allow for movement. This is also going to kind of allow for like folds. This is where the cells that are either secreting things like digestive enzymes or absorbing things, right? The epithelial cells. Okay, so again, stratified squamous starts in your mouth, in your esophagus, and transition to your stomach, small and large intestine. By the time the transition from your rectum to the anal canal is going to go back from a simple columnar to that anal canal, which is going to go back into a stratified squamous. Right? And then again, these gastric pits, gastric juices, gastric enzymes and stuff like that. Big folds for surface expansion, for absorption. These are just the intestinal crypts. I don't really ask any questions on that. All right? Heart. Okay, lecture and practical. You want to name all the vessels, the great vessels coming off the top and then the sides, entering and leaving the heart. The vessels entering are veins. Inferior vena cava, and the superior vena cava, inferior vena cava below, and then the pulmonary veins into the right atrium. Right? So the vessels leaving are be your aorta and your pulmonary trunk. Those are going to your lungs, and then the body. Right? So you should be able to say, okay, which of the following is deoxygenated blood passing through, right? You might say right AV valve or pulmonary valve or something like that, right atrium, something like that, out of a choice of other things that don't have that. So you want to just understand a nice, easy picture of the heart. You could probably draw it by yourself on the sheet. You do that little very simple thing. Just draw a little square. Right atrium, right ventricle, left atrium, left ventricle, entering, leaving, entering, leaving, and then the valves. Right? So you just follow the blood like that from body to lungs. That's why I don't write. I'm too impatient for this. Right. From lungs to body. Right? Once you get this down, it's very simple. Then just name everything. You got the valves over here. Right AV valve, pulmonary valve, left AV valve, aortic valve, right? And what's the function of the valves? Any valve, whether you're talking about semilunar or AV valves, what's the function of the valve? Prevent backflow. That's it, right? They open and close. Why?
Because of pressure.
Pressure differences, right? If it's greater over here, the valve will open, but it'll also close due to pressure, but it's a one-way valve, right? It'll close due to pressure. They'll open and close due to pressure. What's the whole purpose of the papillary muscles and chordae tendineae?
Keep it shut.
To keep it shut, right? They don't open and close them, they just keep it shut because the ventricles are really putting a lot of pressure on. They might prolapse in the valve, the vessels, the blood would go back into the atrium without good chordae tendineae and papillary muscles. All right? Everybody good on the heart?
Yes.
And then also that contraction, it's going to go upper, lower, right? The atrium's contracted once, the ventricle's contracted once. And that is all controlled by that signal that's spreading through, right? The cardiac muscle starting with the SA node. The signal spreads down throughout the atrium, and then the atrium contracts into the ventricles, right there, right? It also reaches that AV node over here. There's a little bit of a delay so that the ventricles aren't contracting at the same time. It's going to go down the bundle branches, okay? When it goes down the bundle branches, it's not contracting, it's still in diastole here. Once it reaches the apex and goes up those Purkinje fibers, does the ventricle start contracting. And that way we get a contraction from the apex upwards, squeezing the blood up into those vessels, right? Make sense, the conduction sequence? SA node, AV node, bundle branches, Purkinje fibers. Okay, just kind of understand what signals are spreading what through what conduction fibers. All right, so circuitry over here on this side, this blue one is the pulmonary what? Artery or vein?
Artery.
Artery, right? Because leaving the heart into the lungs, goes to the capillaries, comes back to the pulmonary veins. For the rest of the body, it's going to be, this is your systemic vein, right? That's a systemic vein, that's your subclavian, and there's a systemic artery. Remember fetal circuitry also, you're bypassing the lungs basically, you're doing that by pumping out from the pulmonary trunk. Is going to go right into the aorta, into the systemic circuit through that ductus arteriosus. All right. Did you do this one?
Mm-hmm.
So on these, just to eliminate ones and then focus on the other ones, you want anything like this, multiple choice, eliminate the ones you know, maybe by looking at the inferior vena cava and the aorta first. Look at the direction of blood flow. That will allow you to hopefully eliminate two off the bat. Which ones for the inferior vena cava are correct? B and C. Okay, so you can eliminate A and D right out. Well, D is also correct, but then the aorta is opposite. So that leaves you with B and C, and then what? And what are these? They're going into the lungs. Right? So these are arteries and veins. Blood's going in, coming back out to the pulmonary veins. Right? Make sense? Okay, and then again, arteries and veins, right, as far as practical and lecture-wise, right? Veins are big, fluffy-ass things, because they have very small tunica media right there. They're under zero pressure, basically. That's why they collapse without fluid in them. Whereas the arteries, you can see, like the celiac trunk, they kind of hold their structure. They have a very thick tunica media. Over here, right? And that reflects their function, the pressure differences. The arteries are your stressed volume, right? That's where all the pressure is. Once they go through the capillary beds, it's basically coming back under zero pressure. We also talked about the different ways blood can go through, One capillary bed, that's the normal thing. Two capillary bed represent what?
Hepatic portal system.
Yeah, in this case, this would be, the hepatic portal system would be, what's an example of an organ containing a second capillary bed? What would be the organ here?
Liver.
The liver, right? This would be the intestines, this would be the liver. This could also be your hypophysis, your hypothalamus and stuff like that. There is like other portal systems. Even a kidney technically would be a portal system, but that's basically, that would be that and that, right? Which one of these, so that's the portal systems, which one of these would depend on precapillary sphincters, you think? What's that?
C.
C, right? Because the precapillary, before the capillaries, if the sphincters close off, then blood won't go through the capillaries. Okay? Remember that. And it'll go through this thoroughfare here. So this is a shunt, right? Shunting blood away from the true capillaries. That would be an arteriovenous anastomosis. Anastomosis. I always remember it by saying there's the good Moses and there's his evil twin, the nasty Moses. That's how I remember it. Right? And then over here you got, again, you can go to this capillary bed, you can go this way, or you can go this way, right? If this one is clogged or shut off for some reason, that's an arterial anastomosis. Two ways of getting to the same place. And you can leave the capillary bed this way, or you can go this way, right? That's a venous anastomosis. Right, like your superficial and your deep veins. That's a venous anastomosis. You can go back to your deep radial and ulnar veins. You can go back to your cephalic or basilic veins, right? That's a venous anastomosis. Right, what's an example of the circle of Willis? Which one of these would represent the
It could be
circle of
D.
Willis?
Arterial.
Delta.
This one, right? Yeah. Arterial, right? That's again, you can go to the front of your brain from the vertebral artery, which normally supplies back to those posterior communicating arteries. All right, for vessels, again, thick arteries, sturdy, thick arteries under pressure. Big, thick tunica media, that's the difference. Right? They're also very expansive. The elasticity, right, will allow that sort of recoil to push the blood forward. Whereas veins, again, big, floppy things, very thin tunica media. The adventitia might be the same, the intima is the same, with the exception of the valve structures. Right? But otherwise, the tunica media is the big difference. Capillaries have lost everything. They're just basically a tunica intima because their whole purpose is to exchange stuff to go in and out of them. So you want to lose all that other stuff. All right. What are the three types of capillaries? And which is the most leaky one? Which is the least leaky one? Which is the middle leaky one? What? Sinusoidal are the most leaky, right? You find those like in your bone marrow and your spleen. And your fenestrated ones, we talked about the glomerulus. That is a fenestrated because you need a lot of fluid coming out of it. And your intestinal capillaries also have that for greater absorption. But your normal capillaries are called continuous. Those are the tightest. Fenestrated are the second leaky, and then sinusoidal are the most leaky. Who picks the interstitial fluid that's built up here? Your lymphatic vessels, right? What else do the lymphatic vessels do? They pick up pathogen. The structures they go through are the lymph nodes, right? The lymph nodes, and then they will mount an immune response. The spleen does the same thing for blood, right? The spleen filters blood. It picks up pathogens and stuff and can mount an immune response, okay? The spleen monitors blood for pathogens. It's part of the lymphatic system. Okay? And then again, remember, greatest pressure. What's the greatest pressure? Your carotid arteries or your posterior tibial artery? The carotid, right? Closer to the heart, basically. Right? As you leave the aorta, as you go farther away, the pressure is going to decrease. This has to do with something physiologically, but whatever. It's going to decrease, but it's going to force blood into the capillaries, but by the time you go through the capillaries, you're coming back under zero pressure almost. So how do we get blood back up this way? What are the three mechanisms? Venous valves, skeletal muscle pump, thoracic pump, everybody understand those? The skeletal muscle pump is you moving around and squeezing blood upwards. The valves catch it. Right, the valves are gonna catch it. And the thoracic pump is for your, like your inferior vena cava, for instance, right? It doesn't have valves for some reason. And when you're breathing out, if your thoracic cavity expands, that means it's putting pressure in your abdominal cavity. And when you increase your pressure here, that's gonna push the pressure, it's gonna bring the blood upward.
What were valves called? Just one-way valves? Just one-way vein?
Venous valves, yeah, they're the same thing as like your semilunar valves, basically. They're just one-way valves that prevent backflow. Alright? So those are the three mechanisms. You lose adventitia when you get to the arterioles, right, and your capillaries, because they're embedded within the organ. And then your elastic arteries have the most amount of elasticity. Remember the green balloon and all that thing, so you got a constant pressure. Everybody remember the green balloon?
Loss of
Fondly.
adventitia at the arterioles?
What's
At
that?
the arterioles, the loss of adventitia?
Yeah, the arterioles will lose because they're basically, these are like outside, these are embedded within the organ. So they'll have the connective tissue of the organ around them. And the capillaries will basically be just your tunica intima.
That's what adventitia is?
Adventitia is the outside, the tunica externa.
Oh, okay.
Right, that's the connective tissue layer on the outside.
Thanks.
All right. Any questions on the blood vessels? All right, so kidneys, everybody understand, like on the models, right? Here's the pyramid, here's the cortex. So here's the pyramid, here's the cortex. Everybody kind of understands the scale thing here, right? So you got 1, 2, 3, 4, 5 pyramids shown here. You'd have the same amount of minor calluses. Basically, each pyramid is associated with a minor callus. There's one ureter. So like how many pyramids observed? How many minor calluses? Like, so if I say, what's the most numerous thing in a kidney, and I give you a list, it's about scale. Right? There's one kidney, there's a whole bunch of pyramids within them, the same amount of minor calluses. Is there more cortical radiate arteries than afferent arterioles? Yeah, because they come off the afferent arteriole. Is there more glomeruluses than afferent arterioles? No, they'd be the same amount, because each one feeds in. Right? So is there more collecting ducts than nephrons? It'd be less, right? Because a bunch of nephrons feed into that. So you should understand those kind of things, because there are those tricky questions like that.
So nephrons are only in the medulla?
No, the nephrons are going to be both. The nephrons start with the glomerulus,
Yeah.
and they proximal convoluted tubule, nephron loop.
Okay.
Distal
Okay,
convoluted tubule is all the nephron.
That makes
So
sense.
here's two nephrons here. They're feeding into a single collecting duct.
Okay.
Okay, that's the functional unit of the kidney is the nephron. This is what's doing, again, absorption over here, reabsorption is the main function of your proximal. Concentration is good enough for a little word. C, concentration, over here. And then the distal one, you could think of secretion will be the most good word for secretion. Okay, good word. All right, everybody understand this? No, I know, you know what this is? This is the amount of fluid filtrate that's made, and this is the amount you pee, which means all the pink stuff is what everything else that happens in the renal corpus. This is all that's reabsorbed here, basically. So in other words, the whole function of the kidney is weird because you're basically dumping a bunch of stuff you didn't want to, and now you have to get it back. That's the whole function of the kidneys. And then also controlling how much you pee out. So you dump out a bunch of stuff, remember, good stuff, bad stuff, right? All your urea, your creatinine, whatever. And that's what you want to excrete, but you've also dumped out a lot of more water than you want to get rid of, a lot of glucose, amino acids. All that's going to get reabsorbed, or most of it's going to get reabsorbed. Okay? Secretion is the process where you want to get rid of stuff that's in the blood, like too many hydrogen ions or toxins. We may as well say, well, let's just pee it out. They can secrete it into these tubes to get rid of it. All right? That's the whole kidney thing. Everybody got the big picture? Can you follow the filtrate as it's made and as it passes through there? What are the functions of each region? Okay? Where's secretion happen? Where do you associate secretion with?
DCT.
DCT, right? Absorption, reabsorption, PCT, filtration, glomerulus, the renal corpuscle, the glomerulus, the renal corpuscle, and the capsule. All right, so that's basically, this picture shows, you know, and again, reabsorption from the tube back into the body, blood. And that's what reabsorption means. Right, because the tube is really, it's kind of like your digestive tube. When you absorb stuff, that's the first time it actually enters into your body. Right, whereas secretion in your digestive system would be like secreting digestive enzymes into the tube. Here, you're secreting other stuff into these tubes. Right? Collecting duct, what's the function? What do you want to think about with the collecting duct? Anybody remember anything about the collecting duct? No. What about alcohol? What does alcohol do?
The ADH.
ADH. Right? ADH, it inhibits ADH. ADH makes the collecting ducts more permeable to water. So when ADH is secreted, your hypothalamus says you're dehydrated, it secretes ADH, and water leaves the tubules, making your urine more concentrated, because it wants to get your water back in your body instead of peeing it out. Whereas if you drink a lot of alcohol or caffeine, then ADH is inhibited and you pee out a lot of water, and then you're dehydrated later. Okay? Makes me thirsty just talking about it, because I have been drinking. Just kidding. People are recording. Urinary system also, you know, will include the ureters, the bladder, the urethra, and the male. Again, you got the prosthetic urethra and the spongy urethra. The female, you just got that short urethra coming from the bladder into the, and then somebody's asking, like, this whole area for the female, the reproductive stuff, whole area is referred to as the vulva. And then the vestibule will be like starting from the clitoris, where they have the labia minora, right, and the urethra and the vagina there. That's just the general names. But reproductive stuff, you got the testes, the epididymis, and then the spermatic cord, right, will contain the vas deferens. Here you don't see it, but it would go around the back over here, and then it would join up with the urinary, the urethra over here. And the prostatic urethra. This over here is what you call the ejaculatory duct, and then it goes through the prostatic urethra and out the spongy urethra. This represents the corpus cavernosa, the erectile bodies of the male penis. Right? And then for female, the ovaries, right? That's the homologue of the testes. Those are the ones. What do? What's the function? Zygote shit. Quite. Zygote is going to be the implantation part, but ovaries are going to make the eggs, the gametes. Testes are going to make the gametes, the sperm, right? And also both of them are going to produce what? What else do they do besides producing the gametes? Their other main function? They are endocrine organs. They're testosterone and estrogen and progesterone, right? Coming from those organs, right? And they're homologous, remember, because they come from the same embryonic plates and they have similar functions. Again, this is a bladder, this is a bladder. Those are not homologous, those are the same things. Okay? These are homologous because they're kind of similar, but they are different. There's no difference. All right? So for where the eggs go, after they ovulate, they pop out of the ovaries, caught by the red thing there, which is the fimbriae, goes into the fallopian tubes or uterine tubes, into the uterus. Remember, the endometrium is where the inside part, where they would implant. Myometrium is the muscle part. Fundus is the anterior or superior part. Cervix is the inferior or posterior part of the fundus. Right? And then the fundus, the cervix meets up with the fornix. Right? That's the cup-shaped region of the vagina, right? And then the vaginal tube and vaginal opening leads up into the vestibule there. Right? Yeah, and then that's just what we went over again. Testes, epididymis, vas deferens, prostate. These are the bulbourethral glands. This is the where the urethra is, that's the spongy urethra, and these are the paired corpus cavernosa. Right, whereas over here, you got the ovaries, fallopian tubes, uterus, you got cervix, vagina, and leading out into the entrance. Again, just gonads. The gonads are the ovaries and testes, and they both produce gametes, they both produce their respective hormones. And then again, the homologs. I love homologs. That's the most interesting thing about this, that we all start out the same, right? But then we change, right, into the meerkat or the alien over there. These are cross sections, the penis and the clitoris here, right? Penis, the glans penis, the clitoris are homologous organs. They come from the same place. They actually have both corpus cavernosa, erectile bodies in them, right? Whereas the penis also has the internal spongy urethra around the urethra, whereas in the female, it's in the outside vestibule. The labia minora and the urethra are kind of the homologs, right? The scrotum, where the testes are would be the homologs for the labia majora, which don't have the ovaries in them, unless the female had descended ovaries, then they'd end up there. All right? So those are the homologs. Be able to match those. Where's the homolog for the uterus? There is none. There's no uterus in men. Men don't have those baby-making things. Do females have a prostate? No, there's no homolog for that.
They don't.
Right, thank you. These are tease questions. You should be able to get this. Again, there's only two female structures here. One of them is the one that makes gametes, right? Uterus is what would be the womb, testes and prostate in the male. Location. Also this, again, fertilization, right? The sperm enters the vagina but goes through the uterine cavity into the fallopian tubes up to the ampulla. So fertilization would take place all the way up in the fallopian tubes typically. Right? Well, that's it. That's all I have.
That's it.